Question 1. Which of the following statements correctly differentiates between gene editing, GE, and genetically modified organisms, GMOs? The correct answer is option C. GE incorporates modification of existing genetic material to improve quality, while GMOs typically involve the insertion of foreign genes, transgenes. Explanation: the text clearly states that gene editing, GE, incorporates modification of existing genetic material to improve quality. In contrast, genetically modified organisms, GMOs, have transgene, foreign genes, for improvement in quality. This is a key distinction in biotechnology. Question 2. Which gene editing technology was primarily used by Indian researchers to develop the first ever low pungent mustard variety? The correct answer is option C, CRISPR-Cas9. Explanation: the news explicitly mentions that the new low pungent mustard variety was produced through gene editing of Varuna using CRISPR-Cas9 gene editing technology. While other technologies like cedefins and T-aliens are also gene editing techniques, CRISPR-Cas9 was the one employed in this specific development. Question 3. What is the canola quality limit for glucosinolate content in parts per million dry weight that the new low pungent mustard variety aims to achieve? The correct answer is option C, 30 ppm. Explanation: the text states that the new mustard variety is less pungent as it has lesser quantity of glucosinolate at par with the canola quality limit, 30 parts per million or ppm dry weight. High glucosinolates can cause health issues in livestock. Question 4. The Genetic Engineering Appraisal Committee, GEAC, responsible for environmental clearance of GM crops in India, is established under which Indian Act? The correct answer is option B, Environmental Protection Act, 1986. Explanation: the text clearly states that the Genetic Engineering Appraisal Committee, GEAC, is established under the Environmental Protection Act 1986. It functions under the Ministry of Environment, Forest, and Climate Change and is crucial for regulating GM crops in India. Question 5. Consider the following statements regarding Bt cotton in India. 1. It is the only genetically modified, GM, crop currently approved for commercial cultivation in India. 2. It contains isolated genes from the soil bacterium Bacillus thuringiensis, which are toxic to bollworm insect pests. 3. It has recently shown increased efficacy against the pink bollworm, PBW, pest. Which of the statements given above are correct? The correct answer is option B, 1 and 2 only. Explanation: Statement 1 is correct. Bt cotton is indeed the only GM crop that has been approved for commercial cultivation in India. Statement 2 is correct. It contains isolated genes from the soil bacterium Bacillus thuringiensis, Bt, and codes for proteins toxic to bollworm insect pests. Statement 3 is incorrect. The text mentions that Bt cotton is said to have lost its efficacy against the pink bollworm, PBW, pest, and GEAC has asked states to test a new kind of transgenic cotton seed with the CRY2AI gene for PBW resistance. Question 6. Dhara mustard hybrid 11, DMH11, is considered a transgenic crop primarily because it contains. The correct answer is option B, genes from a different species artificially inserted into its genome. Explanation: the text states that DMH11 has been developed by Delhi University containing 2 alien genes isolated from a soil bacterium called Bacillus amyloliquefaciens. It explicitly mentions that DMH11 is a transgenic crop because it uses foreign genes from different species. This introduction of foreign genes is the defining characteristic of a transgenic crop. Question 7. Adzima, recently approved by the USFDA, is the world's first recombinant protein product indicated for on-demand enzyme replacement therapy, ERT, to treat which rare blood clotting disorder? The correct answer is option C, congenital thrombotic thrombocytopenic purpura, CTTP. Explanation: the news states that the USDA approved Adzynma, the first recombinant, genetically engineered, protein product indicated for on-demand ERT. It will be used for treating congenital thrombotic thrombocytopenic purpura, CTTP, a rare blood clotting disorder. Question 8. Which of the following statements best describes the primary mechanism by which gene-drive technology, GDT, aims to reduce mosquito populations? The correct answer is option B. It modifies genes to alter Mendelian inheritance, increasing the likelihood of producing sterile offspring. Explanation: the text explains that gene-drive technology, GDT, is a type of genetic engineering technique that modifies genes to alter Mendelian inheritance. Applications based on GDT have shown promising reductions in mosquito populations by making them produce sterile offspring. This is a direct genetic intervention, not an ecological or chemical one. Question 9. Scientists recently achieved the end-to-end sequencing of the Y chromosome. What is the specific term used for this complete sequencing from one end, telomere, to the other? The correct answer is option C, telomere to telomere, T to T, sequencing. Explanation: the text explicitly mentions that the Y chromosome is the last human chromosome to be sequenced end-to-end, telomere to telomere, T to T. Telomeres are protective caps at the ends of chromosomes. This achievement signifies a complete and gapless sequence. Question 10. The Indigen program, which completed whole genome sequencing of 1029 self-declared healthy Indians, was initiated by which organization? The correct answer is option C, Council of Scientific and Industrial Research, CSIR. Explanation: the text states that the CSIR initiated the Indigen program in April 2019, in which whole genome sequencing of 1029 self-declared healthy Indians drawn from across the country has been completed. This distinguishes it from the Genome India project, which is an initiative of the Department of Biotechnology, DBT. Question 11. The world's first baby born using 3 people's DNA through mitochondrial donation treatment, MDT, was recently reported in which country? The correct answer is option C, United Kingdom. Explanation: the news explicitly states that a baby has been born using 3 people's DNA in the UK with help of mitochondrial donation treatment, MDT, procedure. This marks a significant development in reproductive medicine. Question 12. Regarding mitochondrial DNA, metrictin DNA, in humans, which of the following statements is correct? The correct answer is option C, mitochondrial DNA comes only from the mother. Explanation: the text clearly states that mitochondrial DNA comes only from the mother. This maternal inheritance pattern is a fundamental concept in genetics and is the basis for procedures like mitochondrial donation treatment, MDT, which aims to prevent the transmission of mitochondrial diseases from a mother to her offspring. Question 13. Which of the following are potential applications of cell-free DNA, cfDNA, as mentioned in the text? 1. Detecting genetic abnormalities in fetuses. 2. Early detection, diagnosis, and treatment of cancers. 3. Monitoring immune response after organ transplantation. Select the correct answer using the code given below. The correct answer is option D, 1, 2 and 3. Explanation: the text lists all 3 as applications of cfDNA, applications, or detect genetic abnormalities in fetuses, or early detection, diagnosis, and treatment of cancers, or monitor immune response after organ transplantation and can be used as a biomarker. Therefore, all statements are correct. Question 14. What is a primary benefit highlighted for the creation of synthetic human embryo-like structures using stem cells? The correct answer is option C, to understand the impact of genetic disorders and biological reasons behind recurrent miscarriages. Explanation: the text lists several benefits of synthetic embryo, including understanding the impact of genetic disorders and biological reasons behind recurrent miscarriages. These structures provide a model to study early human development without using natural embryos, offering insights into developmental problems. Question 15. The world's first live birth of chimeric monkey was recently reported in which country? The correct answer is option C, China. Explanation: the text explicitly states that world's first live birth of chimeric monkey was reported in China with 2 sets of DNA. A chimera is defined as a single organism composed of cells of more than one distinct genotype. Question 16. NEXCAR19, India's indigenous chimeric antigen receptor T-cell, CAR-T cell, therapy, recently approved by the Central Drugs Standard Control Organization, CDSCO, was developed by which company? The correct answer is option C, ImmunoACT. Explanation: the news states that indigenous NEXCAR19, a CAR-T cell therapy, is developed indigenously in India by ImmunoACT, company incubated at IIT Bombay. This highlights a significant advancement in indigenous medical technology. Question 17. What is a primary function of the Central Drugs Standard Control Organization, CDSCO, as mentioned in the context of drug regulation in India? The correct answer is option B, to regulate the import of drugs and approve new drugs and clinical trials. Explanation: the text specifies that CDSCO is the central drug authority for discharging functions assigned to the central government under the Drugs and Cosmetics Act 1940. Its major functions include regulatory control over import of drugs, approval of new drugs and clinical trials, etc. Question 18. Cytokine release syndrome, CRS, is a significant challenge associated with CAR-T cell therapy. Which of the following best describes CRS? The correct answer is option C. It is an excessive release of cytokines into the bloodstream, triggering an intensified immune response that can damage organs. Explanation: the text explains that CRS means abundance release of cytokines into the bloodstream, triggering an intensified immune system response. It further clarifies that excessive cytokine production causes an immune response that can damage organs, especially the lungs and kidneys, and even lead to death. This is a critical side effect of CAR-T cell therapy. Question 19. The Diverse Epigenetic Epidemiology Partnership, DEEP, project involving CSIR Center for Cellular and Molecular Biology, CCMB, primarily aims to address which of the following issues in global health research? The correct answer is option C, to generate genomic datasets in underrepresented populations to understand non-communicable diseases, NCDs. Explanation: the rationale for the DEEP project is that much of the population health research conducted till date has drawn heavily on data collected from people of white European origins. Many global communities are underrepresented in health studies. Its significance is to understand the genetics behind non-communicable diseases, NCDs, in diverse populations. This directly aligns with option C. Question 20. Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work, without changing the DNA sequence. One common epigenetic modification mentioned in the text is The correct answer is option C, DNA methylation. Explanation: the text defines epigenetics and then states, one common epigenetic modification is DNA methylation. It further explains that this involves the addition of a methyl group to the DNA molecule and high levels of DNA methylation lead to gene silencing.